words you read, the colors you feel, that magnificent view creates a memory in our brain. Do you wonder how the world is perceived by our brains? Every color, every detail, every movement, it all starts with a tiny, intricate structure at the back of our eyes. In this video, we're going to embark on a journey into the microscopic world of the retina, unraveling its secrets and understanding how it transforms light into vision. As light rays penetrate the eyeball, they dance through the cornea, bending and refracting with precision. It crosses various structures of the eyes, including vitreous fluid chamber. Each flicker of light photons tells a story, capturing fleeting moments and breathtaking detail. These photons finally reach the innermost layer of the retina, the inner limiting layer, or ILM. ILM is formed by the end feet of the molar cells. It is primarily composed of collagen and other extracellular matrix components, which contribute to its structural integrity. It acts as a boundary between the retina and gel-like vitreous humor. Once the photons reach the ILM, it crosses 10 different layers of retina. To reach the retinal pigment epithelium, RPE. The RPE forms a critical part of the outer blood-retinal barrier and selectively transports nutrients from the choriocapillaris. The RPE contains melanin granules, which absorb stray light that passes through the photoreceptor layer. This prevents light scattering, noise, and improves visual acuity. The RPE plays a vital role in the vitamin A metabolism isomerization of retinol, which is essential for regenerating the visual pigment rhodopsin in rods, conopsins in cones. The RPE plays a role in regulating the immune response in the retina, helping to protect it from inflammation and infection. RPE will cause phagocytosis of photoreceptor outer segments. Once the stray noise photons are filtered by RPE, photon of light strikes rhodopsin in rods or conopsins in cones. These rods and cones forms the photoreceptor layer. Now that the photons have triggered the rods and cones, this is where the light photons are converted into neural signals, which later is sensed by the brain as information. The story gets more interesting. You wonder, how does this happen? Let us understand the visual cycle and phototransduction. In resting state, retinal molecule 11-cis retinal bound to the opsin in cones and rhodopsin in rods. When a photon strikes retinal, it undergoes a change in shape, converting from 11-cis retinal to all transretinal. This change triggers a detachment of opsin from retinal. Opsin later causes activation of transducin and leads to phototransduction, which we discuss more detail in a bit. All transretinal which is formed has to convert it back into 11-cis retinal, and this happens by reuptake of it by RPE. It is then stored within RPE, ready to be available for next cycle. This is called visual cycle.
In the dark state, light-sensitive opsin remains attached with the 11-cis retinol. Opsin being inactive and transducin being inactive too. There is plenty of cGMP. cGMP keeps the sodium channel to remain open. This allows more Na ions inside photoreceptor cells. Increased Na ions inside the cell will increase the positivity, that is depolarized state. Depolarized state will lead to efflux of glutamate through its glutamate receptor. This glutamate will then pass on to the bipolar cell, which is synapsed with photoreceptor cells.
In dark, photoreceptors are depolarized. It sends a positive signal to bipolar cell, which is further processed and sent to the brain. Brain perceives it as dark. Light activates opsin-ribopsin. It triggers a conformational change that allows ribopsin to activate transducin. This leads to coupling of transducin with its G protein coupled receptor. Once after coupling, the alpha subunit exchanges GDP for GTP, and then goes on to activate PDE. PDE hydrolyzes cyclic guanosine monophosphate cGMP, reducing its concentration within the photoreceptor cell. Low cGMP causes closure of the sodium channels, and hence more negativity inside photoreceptor cells. Opposite signal is perceived by the bipolar cell in the absence of glutamate. In light, photoreceptors are hyperpolarized. It sends a negative signal to bipolar cell, which is further processed and sent to the brain. Brain perceives it as bright. Photoreceptors helps to pick the light and perceive it as switch on and switch off signals, like a binary language, but with complexities of many gray and colors in between.
To understand better, we shall initially focus on only black and white interpretation. If you consider each rod cell representing each small boxes in this picture, the level of darkness and brightness is merely captured by a rod cell based upon its different strengths of hyperpolarization. Higher the hyperpolarization, brighter the box. Higher the depolarization, darker the box. Complete vision is a very complex process where environment is perceived by retina have various combinations of colors, brightness, contrast effect, size and depth of the field, etc.